Small signaling molecules target various effector proteins to control bacterial virulence and human biology. However, these effector proteins are challenging to identify. As a systems biology tool, DRaCALA allows a feasible, rapid, and highly sensitive identification of the unknown effector proteins by using an ORFeome library.
DRaCALA could be used to study any small signaling molecule as long as it can be labeled with either radioactive isotope or fluorescent dyes.
Start by inoculating E. coli into 1.5 milliliters of Lb supplemented with 25 micrograms per milliliter chloramphenicol in each well of 96 well deep well plates for an overnight incubation at 30 degrees Celsius and 160 rotations per minute. The next morning, treat the overnight cultures with 500 micromolar IPDG for 6 hours at 30 degrees Celsius to induce protein expression. At the end of the incubation, pellet the cells by centrifugation, remove the supernatant, and freeze the samples at -80 degrees Celsius. To initiate lysis, resuspend the pellets in 150 microliters of lysis buffer 1 per well for a 30-minute incubation at -80 degrees Celsius before thawing the cells for 20 minutes at 37 degrees Celsius. The lysate should then be stored at -80 degrees Celsius before use. To complete the lysis of cells with overexpressed proteins, resuspend the samples in 40 milliliters of ice-cold lysis buffer 2 and sonicate the samples at a 60% amplitude at 2 seconds.